I've known Richard since he started graduate school at MIT. But before I tell you about that, let me go back. He was born in Nairobi, Kenya, and did his undergraduate work at Trinity College in Cambridge, where he got first-class honors in biochemistry. For those of you who don't know, that's where Isaac Newton went to school, and is also the site of the famous Christopher Wren Library, so it's well worth a visit.
Newton was no longer there, so.
Yes, we know that. Yeah. Nor was Nehru. But then he had the good sense not to go to graduate school in England, but come here to MIT, where he was a student with Paul Gross, which is, I think, when I first met him, but I'm not really sure of that, because he was just a graduate student then. And then returned to England, where he was a postdoc at the ICRF in London, where it's notable because it was there that he identified fibronectin as a cell surface protein that was very important in oncologic transformation. And that really set his career in motion. He came back to MIT as an assistant professor in 1975. And I'm sure I'll tell you all this, but he worked out a lot of the biology of fibronectin, its receptors, the integrin signaling pathways, studied a number, in fact, cloned a number of important cytoskeletal and membrane proteins, and he'll tell you all that. He's been very important to the life of the biology department at MIT. He was associate head of the biology department from 85 to 89, which would be under Maury Fox, right, if I'm right. And then took over as head of the biology department for just a couple of years, and then switched jobs with Phil Sharp. So Phil became head of the biology department, and Richard took over Phil's job as head of the cancer center. Why that happened, I've never figured out, but anyway, it's been great to have him. He'll tell you that in both jobs. Currently, he's a Hughes investigator, the Daniel K. Ludwig Professor for Cancer Research at the David H. Koch Institute for Integrative Cancer Research, Department of Biology, MIT. He's got lots of honors, just a couple of them I can mention. He's both a fellow of the Royal Society of London and a member of the US National Academy. He's won a number of awards, most recently the E.B. Wilson Award, which is the highest award given for the American Society for Cell Biology, which he also served as president. And I think I've gone on enough. Richard, welcome to MIT.
Thank you. For the third time, I guess. So. Well, thank you all for coming. You should interrupt and ask questions, but if you don't, I'll keep talking. So, I guess I'm supposed to tell you how I got here and why I got into biology, what motivated the career that I've had. And so I guess I didn't really have very much choice in the matter, because both my parents were scientists. My father was an entomologist and a freshwater ecologist. And all of the time I was growing up, he was doing experiments in the back garden and in his lab at Liverpool University. And my mother was a physicist and taught college physics in I guess you'd call it a junior college here. So when I or my siblings, there were 4 of us, used to ask questions, we never got answers, we got questions, which led us to answers sometimes. And if we looked for help with our homework, it was the same thing. So we were sort of brought up to be scientifically inclined. And when we went on family vacations, my father always had his bug net, and any stream or pond we came to, he'd be collecting stuff out of it and telling us about it. And we got to be able to identify all sorts of aquatic insects and their larvae and so forth. And he was an ecologist and became one of the early experts on how you study polluted waters, of which there were a lot in northern England, it being an industrial part of the country, and all the rivers had been polluted. And so he studied how do you find out how things are polluted and how they get better. And so we grew up with this all around us. And in fact, all 4 of us became scientists. So I guess we were effectively brainwashed. So not kicking and screaming. I guess if we'd not wanted to be, we'd have changed tack. But I always did want to be a scientist from the very beginning. And in England at that time, I guess still, but slightly differently, one went to high school at the age of 11. There was primary school and then there was high school. And at that age, you could choose a stream. You could be either modern languages, classics, or science. And it didn't mean you did nothing else, but it meant you had a bias towards science. So I naturally chose science, having been, as I said, brainwashed already by then. And for, I guess it's 4 years, that's what I did. And then we all took a set of exams in a broad spectrum of subjects, but if you were in the science stream, enriched in science. And we used to do a lot of lab work. I think it was 2 afternoons a week. All those 4 years we were in the lab doing physics or chemistry, not biology. Biology was sort of looked down upon as not being a serious science in those days. You did biology if you wanted to go to med school. So I didn't want to go to med school. I never had any inclination to go to med school, but I did want to do biology. And I used to get biology information at home from my father, who had things like New Biology and New Scientist around at home, and I used to read them, discuss things with him, and go to his lab and play around with the various bits and pieces there. And one summer when I was in high school, I had a job in the School of Tropical Medicine in Liverpool, which was still is a big tropical medicine school. You know, there used to be colonies, like Kenya, that had tropical diseases, and so there were a lot of tropical medicine schools. And I had a summer job working on mosquito emergence. There was this idea that if you dropped pellets of high long-chain alcohols into all the bits of standing water, they'd make a monolayer on the surface, and the mosquito larvae would not be able to emerge through the surface because the surface tension would be altered. So that was my job, was to test that out, which I did. It didn't work. A fairly conclusive set of experiments that it made no difference to mosquito emergence. So I spent most of that summer in a room in the basement, 90 degrees, 90% humidity, being bitten by mosquitoes. So I used to feed the mosquitoes a chicken every night. They put it in the cage and it would get bitten all night, and then they'd lay their eggs and I'd put them in the test, and I got bitten a lot. And it's actually had a good consequence. So at the beginning of every season, I get bitten and a few bumps come up, and then they all go away and I never get any more bumps. And Hermann Eisen tells me that this is because I was heavily immunized when I was this high school student and induced an IgG response, which comes back every year and takes over from the IgE response, so I don't get inflamed mosquito bites. So if you have mosquito bite problems, the thing to do is overdose, and then you may have the same result that I did. Anyway, the teaching was really good at this school. It was a big city high school in Liverpool. And in the last 2 years of school, what's called the sixth form in England, you got to specialize really. And basically we did science 80% of the time, and some modern languages and a bit of current affairs and some literature, and that was it. So for the last 2 years at school, I spent 3 afternoons a week in the lab. Now you could do biology. As I said, it wasn't, you only did that if you were going to medical school, but I wanted to do it anyway. So I did all 4, math, physics, chemistry, and biology, and it was a ball. We had these labs every week. We'd spend 3 hours in the lab, and we did everything from making our own histological stains, staining tissues, sectioning things. And I just loved it. It was so much fun doing experiments. So I decided to go to Cambridge, which was the place to go to do biology in those days, and applied and got in. And because of a quirk in the English system, you kind of get into Cambridge or Oxford by Christmas, and you can't go till the next fall. So I was beginning to waste time and play cards and cause trouble. So my parents decided they should send me away to do something more interesting than waste my time for 8 months. So my father had a friend at UMass Amherst, another entomologist. So I went to UMass Amherst for a semester to experience America. So I took a lot of courses, and they were chosen mostly to leave Tuesdays and Thursdays free so I could ski with this entomologist who was a skier. So I learned to ski, and I also did some really great courses. It was a really good experience. The style of teaching was different here. I prefer the style there, but it was different, and I learned a lot from it. I took a great course from this entomologist, which was evolution from the Big Bang to man. The whole semester taught at the sort of scientific American level. And I've been interested in evolution ever since then, actually. That really stimulated something which has entertained me ever since. And I took a comparative physiology course and thermodynamics and some other things. And I had a great time. And then I went back to England, spent the summer in Liverpool, and then went to Cambridge. And as I said, Cambridge was the place at that time in Britain. Maybe, certainly in Europe, maybe it was one of the best places in the world for doing what we'd now call molecular biology. It wasn't a course in molecular biology, then it was called biochemistry, but it was really biochemistry and molecular biology. So the way the British system works is you specialize completely. You don't do anything else except science if you're a science major. So I did broad sciences for 2 years and discovered I was better at biology than I was at physics, so I stuck with that. And then the final year, what's called part 2, is entirely the subject you major in. And we had to come back the summer before and do experiments in the lab for 6 weeks. And then during the rest of the academic year, we had nothing but biochemistry and molecular biology. Sort of a lecture at the beginning and the end of every day and labs in between. So I spent 4 days a week in the lab doing experiments, learning how to do experiments. And the end of it was a sort of project for a month where you worked with a TA. My TA was Tim Hunt. And he was, at that point, working not on the cell cycle, but on protein synthesis. And the project was to figure out how many ribosomes there were in a polysome, which had just been discovered here, actually, in Alex Ritchie's lab. So we were working on that, and I spent a lot of time in the cold room with sucrose solutions. It was lots of fun. Tim was a very inspiring TA. So the teaching, I'd say this is probably the thing that I would look back on most of everywhere I've been, is that the teaching's been fantastic. Really challenging in all the places, from high school to Cambridge to MIT. I'll come to that in a minute. To maintain one's interest in finding out things. I'll say a bit more about that when I come to MIT. So the other thing about Cambridge teaching is you have what are called tutorials in addition to the lectures. The lectures are ex cathedra. People don't ask questions. They're often big performances, and they're very good, but it's not the sort of byplay that you have in a lecture here. So that's done in what are called tutorials with 2 to 4 students, sometimes even only one, and a teacher who's usually a grad student or a postdoc or a junior faculty member. And those were a required part of the courses, and we had to write essays for them. And I remember we used to go out to the laboratory for molecular biology at Hills Road, the place where Crick and Brenner and Perutz and Kendrew, et cetera, later Bob Horvitz, were, and it was the sort of the temple of molecular biology. And we used to go out there for our tutorials. And I remember one of the tutorials was on a preprint of the wobble hypothesis. This is at the time when the code was just getting worked out, hadn't quite been worked out. And there was this problem about how do you deal with the degeneracy of the code. And Crick had written this theoretical paper on the wobble hypothesis, about the third base not recognizing, being redundant and variable in what it could recognize. So I had to give a presentation on that to the biochemistry department. And they were learning about this for the first time through the student presentation, which was kind of cool. And so it felt like what later MIT felt like, that the students were up there with the faculty, and there was a dialogue in which everybody participated. And it's one of the things I really like about MIT as well, that it's not hierarchical, that anybody who says anything intelligent gets a hearing, and anybody who says anything stupid, even if they're faculty members, get called on it. And I think that's the way it should be. And it was the way it was in Cambridge, and it was, and I think still is, the way it is here. And it's something that it's up to you students to maintain. Stick your oar in. Even if you think you don't quite know, you probably have a better idea about the question than you think you do. So it's good to ask it. So coming to the end of Cambridge, college was only 3 years in Britain in those days. Well, still is, actually. I had to decide what to do, and I didn't want to stay in Cambridge because it's a small town, it's a bit insular. And I wanted to do eukaryotic biology because I'd been interested in that from school days when I was reading about the code and ribosomes and polysomes and all that stuff. There was also going on at the same time the understanding of muscle contraction, how the sliding filament model could explain how your muscles could contract. And that went right down to the level of individual molecules. You could see myosin molecules in the electron microscope. You could see them assembling into filaments. You could see those filaments in the muscle, and they were sliding. And I thought this was really cool, that you could understand a macro event like a muscle contraction in terms of molecules. And similarly, there was a lot of plant physiology being done at the time, and why do plants grow up towards light and down towards gravity? And this molecule indole acetic acid had just been discovered, and it was clear that that molecule was controlling a lot of these things in response to gravity or light or turgor pressure. So I got really, and still am, enthused by the idea that you could understand big things in terms of molecules. It was coming about at that point. That was molecular biology, I guess, but it had ramifications beyond the code and all of that stuff. So I wanted to do something more complex than the code. So I was looking around for places to go, and my tutor at Trinity, Brian Hartley, who was a protein chemist responsible for sequencing chymotrypsin and working out a lot of its enzyme mechanism, he used to have evening tutorials with all the Trinity people who were majors in biochemistry, and we used to have beer and talk about papers. And so I asked him what to do, and he said, well, why don't you go to the States? There's a lot of stuff going there. He said, why don't you go to MIT? I said, what's MIT? And he said, well, he told me what it was. And he said, they're building a department there that's getting into eukaryotic biology. And I know Vernon Ingram, who used to be in Cambridge. You should apply there. So I did, and I applied to Harvard and Yale as well. So I wasn't sure where I'd get in. Harvard turned me down, so I didn't go there. And Yale and MIT accepted me, and MIT said they'd pay me for a year. So I decided, well, that was good. So I came with the intention, I think, really of working with Vernon Ingram. And I even had a project in mind, because he was then doing both RNA sequencing and protein sequencing. And it wasn't clear how you began and ended reading a gene at that point. And I wanted to sequence the ends of this virus, which is a very simple plant virus I'd come up with, and sequence the ends of its proteins and figure out what it was that defined the ends. Well, when I got here, it turned out Vernon was going on sabbatical, and I couldn't work with him, so I didn't do that project. And I worked with Paul Gross, as Harvey said. I'll tell you a bit about that in a minute. How are we doing? Let me say a bit about the courses first. At that time, MIT's biology department was about 1/3 to 1/4 of the size, right, Gene? Something like that of what it is now. Gene was here in the biochemistry division. The department had divisions. Even though it was smaller, it was divided into divisions. And there was a biochemistry division, and Gene was in that, as was Vernon Ingram. And that was where I was placed. So a lot of the students, I'd say the majority of the students, didn't have a biology background. They came from physics and chemistry and mathematics and divisions like that. So one of the tasks of the department was to teach us some biology. And I knew some biology, but most of the others didn't, actually. And there were several ways we learned it. One was a great course, which I think of as the sort of precursor of 750, which was a course that was given in the first year in the evenings, and it was The Great Questions in Biology. And it was run by Paul Gross and I forget who else. And it was a fantastic course. It was a series of issues that ought to be solved, like, do mitochondria have their own genomes? Is the DNA in the chromosome continuous? How many DNAs are there in a chromosome? Is it unaneme or polyneme? And what are Balbiani rings and puffs in Drosophila chromosomes? All sorts of big questions. Oh, I remember another one, which was protozoa. This one's still not solved, really. Protozoa have these arrays of cilia in their cuticles by which they swim, as you know, Euglena and so forth. And they're very organized. And if you take out a piece of cuticle and turn it around, it retains its organization, and it reproduces its organization. So how is that cytoplasmic organization inherited? We still don't really understand that. So we used to meet every week at night and talk about papers that bore on these questions. And then in the fall, at the beginning of our second year, we had the first part of the prelims. Prelims were a much bigger deal then than they are now. And the first part was to see if we knew any biology by then. So that was a great course. It was a discussion course like 750. How do you find out? What are the questions? How would you find out what the answers are? And it was prescient. A lot of the questions that were posed in that course got answered over the next 10 or 20 years. Some of them, as I say, didn't, and still there. There were some other courses. There was a lot of phage genetics. There was a great course taught by Cy Leventhal and Ethan Signer on phage genetics. And Harvard at the time was more prominent up the road than we were then. And we used to go up to Harvard, and I took courses with Jim Watson and Wally Gilbert and Frank Westheimer in physical chemistry, physical biochemistry. And Charles Tamford was on sabbatical at Harvard that year, and he taught a course on the biophysics of macromolecules. It was probably the best course I've ever taken. Sort of, there were 5-minute periods where I actually thought I understood X-ray crystallography, but they'd sort of diffuse away. But it was a very stimulating course, and he was fascinated by the antibody question. How do you make antibodies? So Sumo talked about this last time. At that time, it was not solved. And Lisa Steiner was coming to the department, and I nearly worked with Lisa because I was so turned on by this question of how do you make antibody diversity, and she was working on that sort of question. But in the end, I decided to work with Paul Gross, who's a developmental biologist, and I got fascinated by that. I'm kind of easily distracted, and I got fascinated by that too. So he was working on early sea urchin embryos, and he had discovered maternal message shortly before that time. And I got fascinated by the question about whether maternal message was segregated to different places in the embryo to endow them with different properties. This is a question that is well studied now. At that time, it was well studied biologically, but there was essentially no molecular understanding of it. There were, quote, determinants, cytoplasmic determinants. There was quite good evidence that you could stir, particularly Ascidian eggs was one good example, where you could move around the colored cytoplasms and change the fate of the cells by which bits of cytoplasm they got. So I was really intrigued by that problem. So I took as my thesis project to find out whether the micromeres of the sea urchin embryo, which are the precursors of the cytoskeleton, of the skeleton, and they're kind of the analog of the organizer in the frog embryo. If you move them around, you'll induce a new axis in the embryo. And they segregate off at an early cleavage, and they're clearly different in their properties. And the question was, well, do they have different maternal messages or not? So this was actually an overly ambitious project, which I never solved. But I worked out ways of separating the 3 different cell types in biochemical quantities so I could address it. And then I started looking at the RNAs, and the technology just wasn't there to answer the question. But I did a lot of COT curves and looking at the complexity of the RNA, and it turns out some of these cells will make cilia and some of them won't, so we did a lot of work on microtubule and tubulin synthesis. Tubulin had just been discovered as the I'm showing my age here, as the subunit of microtubules, a fact you probably got in primary school.
I realize people who don't know what a COT curve is either.
Well, they do if they've taken 750. We still insist on telling them. So. It was a way of measuring the complexity of RNA or DNA by rehybridization. And actually, I had my own technique for doing COT curves. COT curves involve binding DNA to hydroxyapatite, ground up bone, basically, and then eluting it at different salt concentrations for the single-stranded and the double-stranded. And this meant running a lot of columns, and it was boring. So we had a we had a hand centrifuge in the lab that was used for spinning down sea urchin eggs and embryos. And I used it for cot curves because it could spin down the hydroxyapatite in 10 seconds. So I had all these reactions running in a big 60-degree water bath. And I'd take them out, spin them down, take the soup off, and put them back in again. They didn't have time to cool down. So I could run dozens of columns at a time by spinning them down in this hand centrifuge and eluting things. So anyway, it was a lot of fun. The lab was great. We were up on the 7th floor of Building 56, and Cy Leventhal was there, and Botstein showed up at a later stage. Alex Rich was there. And it was a very lively place. Harvey actually arrived after me. Harvey and David Baltimore and John King all arrived around the same time, 67, 68, sometime around there.
Fall 68.
Yeah. So as you probably know, 68 was an incredible year. And as another thing that this has nothing to do with science, has affected my attitude to life ever since. I mean, MIT, like everywhere else, was in revolt. Paul Gross, my advisor, was one of the more conservative members of the department. He particularly disliked the breakdown of the academic structure and the degrading of grades. But mostly this was driven by the war, and we all spent a lot of time marching in demonstrations. And I was watching a film recently about 68, and it sort of took me back to that time and the anger that everybody felt about the war, and that I could transplant to the anger that many of us feel now about the war. And it was actually very comparable. You know, the administration was lying to us then, they're lying to us now, so I'll get back on the point. So, but that was very exciting. It was part of the experience of being here at the time. So I finished my degree, not having answered the question, but having had a lot of fun and done a lot of other things that I hadn't intended. Learned a great deal from the people here. And it had always been my intention just to come to the States, to experience the States, and then go back to England. So that's what we did. We went back. So I asked around the department what to do. And I think particularly Vernon Ingram and Howard Green, who was then a faculty member, said, well, you should go to ICRF in London. So that was the Imperial Cancer Research Fund in Lincoln's Inn Fields in London. It's still there, changed its name. But it was a mecca for doing cancer research in the early 70s. And I mentioned that I'd taken this course with Jim Watson, which was on viruses, both bacterial viruses and the early tumor viruses like polyoma and SV40. And that had much influenced me. And I decided I wanted to go there to do biochemistry on how polyoma transformed cells. It was one of the model viruses. So I went to Michael Stoker's lab, who was an expert on polyoma transformation. Susumu mentioned polyoma transformation last time when he talked. And it was a great system that transformed cells with one virus with very few genes. And it seemed to me that one ought to be able to work out how it did it. How did it actually transform cells? So Stoker had a system for doing what was called abortive transformation, which was the early steps where one might be able to actually to figure out what was going on. So I went there. Turned out that was also intractable. There weren't enough cells to do any biochemistry. So I quested around for something else to do. And at that time, early 70s, the membranes were just becoming sort of understood. The fluid mosaic model had just been published, or slightly after I got to London, by Singer and Nicholson. And there was some idea that you could understand what red cell membranes at least were made of. Well, it was beginning to be understood then. Nothing about tumor cell or normal cells, but I decided, okay, here's another question that's really cool. How do these cells differ? There were some reasons for thinking they might differ in their surfaces, and the techniques coming from red cells might actually work on transformed cells in culture. So I set about trying to purify membranes, and I got really depressed. When you start to be a postdoc, you're quite good at what you do as a graduate student. You go to a new lab and you do something different, and you're a klutz. You can't do anything right. You don't know anything. And so I floundered about for several months purifying membranes, which turns out to be really hard to do, actually. You can enrich for them, but you can't purify them, and I could see I wasn't getting what I wanted. So I started looking for some other ways of doing it, and there was a reagent called Brecher's reagent. There was a non-permeable reagent that you could label the surfaces of red cells and identify everything on the surface because it only reacted with the things it could get to, and it was charged, it couldn't cross the membrane. So I decided I would make some. You couldn't buy it. So the way you made this was you bought 5 millicuries of methionine from Amersham, and you did a series of chemical reactions on it and purified the products on paper electrophoresis. So I did this on Lionel Crawford's paper electrophoresis apparatus. So I wound up with 2 millicuries of radioactive cooling fluid that diffused, the side products that diffused off into the Varsol. And so it was about 2 millicuries of that, and about 1.5 millicuries of the reagent. So they weren't very pleased with me at ICRF because they had to buy new electrophoresis apparatus because it was all radioactive. And the Varsol had to go up on the roof and evaporate before they could get rid of it. So I gave that one up, actually. The reagent didn't work very well on cultured cells because it required a pH above 8, and that kind of made the cells leaky. So then I switched to another technique, which had also been worked out on red cells by David Phillips, which was lactoperoxidase iodination, which is still in use, and got that to work on animal cells, and it labeled lots of bands, and it was great, and there was no difference between the normal and transformed cells until I was browsing through what's going wrong here? Why can't I see anything? And I realized that all the normal cells had a big plug of iodine on the top of the gel, and most of the tumor cells didn't. And so anybody in my lab will tell you, I hate it when they cut the stacking gels off their gels. Because if I'd cut the stacking gels off my gels, I'd never have seen this. Because I was running high percentage gels because all the virologists who'd invented slab gels right around then had small proteins, so they ran high percentage gels. This was a big protein, and it didn't go into their gels. So the next day I ran a 4%, 6% gel, and there was this great big band. I mean, it was just spectacular. There was this band in all the normal cells, and it was either not there or almost gone in all the tumor cells. It's one of those experiments that you never forget, because there was this great big band there, and it was huge. It was 250 kilodaltons. So at the time, people didn't really believe. Some people didn't believe proteins could be that big. It's remarkable. I used to get that for a couple of years. How do you know it's a single protein, not an aggregate? So these were SDS gels. So that was fibronectin. So I started iodinating any cell I could get hold of. And at the time, we didn't have any fume hoods. And so I was doing this on the open bench. So I had a bench here where I'd do the iodinations, and I had a microscope, and then we'd do the gels there. And me and my technician would work there. And when I first started to do this, I looked up iodine. There was no Google then, so I went to the library and looked up iodine and realized it was a gamma emitter. So I told the radiation protection people, I need a gamma counter. And they said, oh, look, the Geiger counter works. And it does. If you stick it near iodine, the Geiger counter works because there's some secondary beta radiation that shows up in the Geiger counter. They wouldn't get me a gamma counter. So I decided I better do something about this. So I bought seaweed pills, and every day I did experiments. I'd take seaweed pills in the morning, and they were pretty awful because they stay with you all day, right? They're kind of like pastrami. You burp seaweed all day. Right? But it turned out it was a good thing, because sometime later they eventually got me one of these handheld gamma counters, and I stuck it up to my thyroid, and it started to tick, right? So I stuck it out the window, and it was still ticking a little bit, but it was definitely ticking more when I stuck it on my thyroid. So they panicked at that point, because I had told them months before that we should do something about it. So they shipped me off to Amersham, me and my technician, who had rather... And we sat in what you guys sit in now if you do any iodine experiments, a big sort of thyroid monitor. And the guys at Amersham just laughed. They said, you've got 8 nanocuries. It's nothing. People get that much from the drinking water in London. So they shipped me back to ICRF, but by then they'd panicked. So they built a whole isotope suite with a fume hood, especially for us to carry on doing these experiments. And we had color-coded lab coats, so we couldn't come out of the room without taking them off. They were bright orange, so that we'd alert anybody. And we had to wear gloves and monitor our hands. So they spent a lot of money. But they wouldn't let me trap the iodine. What I'd been doing in the past had been trapping it in an Erlenmeyer flask with an aspirator, with a lot of reducing agents in there to stop it from going to I2 and becoming volatile. It was working very well. It stayed in there. It was behind a lead wall, so it wasn't a radiation hazard. It wasn't an ingestion hazard, not much anyway. But they wouldn't let me do that. They thought it was dangerous. So they said, you've got to aspirate it straight into the sewers. And actually, the sewers empty into the Thames, and some of the drinking water in London comes from the Thames. So now you know why the people in London have more than I got from my experiments. So. And when I came to MIT, they were similarly very touchy about this iodine, and we had a special dedicated hood, and they monitored the stack, so we couldn't put more than a microcurie up the hood every day. So for months, maybe a year or more, we were the major user of iodine at MIT. But none of us ever got a significant contamination. So this is my little homily, which the lab has also heard, that radiation is not as dangerous as toluene and xylene and histology. So if you take appropriate precautions. Anyway, I had fun iodinating everything. So there's a postdoc there called John Smart who worked on slime molds. Harvey used to work on slime molds. And there was a developmental sequence that was very well described in slime molds, and they aggregated. So they had surface proteins that changed during this developmental sequence. And we decided we would figure out what they were. So we iodinated slime molds every 2 hours during their time course, and actually published the first paper on the surface proteins of dictyostelium during aggregation. And this was a great experiment because it had 2-hour time points, and there was a pub just behind the lab. So we could go until closing time, go to the pub between time points. And then after that, we had to stay sober enough to collect the rest of the time points through the night. So that led to fibronectin, and this was one of the early molecular changes in tumor cells, and we got very excited by it and started working on all sorts of aspects of it. And Salva Luria at that point was just setting up the cancer center, and he came to ICRF to scout talent. And I knew him from when I was here. He was a faculty member when I was a graduate student. He said, oh, we need a developmental biologist, forgetting that by then was no longer a developmental biologist. But we need developmental biology in the center. That was definitely one of his missions. That was why I was hired. It was why David Hausman was hired. Actually, it was a prescient thing. He was right. Although neither of us really did developmental biology. But anyway, he suggested I should come and interview, and I was lucky enough to get offered the job and came to the cancer center shortly after it opened. I was a semester late because my visa wouldn't come through, but I was here early on. And the cancer center was also a great place to be. It was a new place. Salva and David Baltimore had set it up. Baltimore got the Nobel Prize shortly afterwards. Phil Sharp discovered splicing a little bit later. And it was a very dynamic and exciting place, and was a great place to be a faculty member. And I actually came back and taught developmental biology for 10 years with Mary Lou, if you remember. And as I said, one of the reasons I wanted to come back to MIT was because of the great time I had as a graduate student. And wanting to get back to that. The politics had changed. It wasn't as lively politically when we came back. But the science hadn't, and it was still just as lively. I vacillated for a while, actually, because ICRF wanted me to stay. And the Cancer Center had offered me a job, and David Baltimore came to London during that time when I was trying to make up my mind to, in part, and I'm sure that wasn't why he came, but he came to ICRF to see if he could persuade me to come. We went to the Rocky Horror Picture Show. So recruiting was a little different in those days, too. So he and Alice and several of us from the lab went to the theatre in London. Anyway, I came back, and I still think that this place has the right combination of teaching and research. In my mind, they're inseparable. And if you're not teaching or studying things outside what you actually do, you get narrow. And that's what happens in places like ICRF, which was a great place. But everybody was thinking about cancer. And they weren't thinking about development. It changed a bit later, but that was the case then. So it was good for a while, but then when that field is not the most exciting field anymore, it's harder to make the jump. Whereas here, it's very easy to make the switch if you see something else interesting coming along. I think of it as surfing. If you have some big question in your mind about that you'd like to answer, and keep your eye open for how other people are approaching their problems, then you can sort of grab onto the wave when it comes and use it to address your problem. And a place like this, that's very easy to do because it's very plugged into the grapevine. And lots of people around doing interesting things that you suddenly realize are applicable to your problem and you can apply them. I think you have to have your own big problem in mind about what it is you'd like to answer. You don't want to be driven by the technology, but you want to be able to grab the technology as soon as it comes. And I think teaching helps a lot with that because you look up from the bench and you look around at what you're doing and what other people are doing and how it fits in the big picture. Something you don't have to do quite so much when you're working at the bench. You can focus on the daily and the weekly and the monthly and the competition. And you need to keep your head up as well some of the time. So I think that's the great thing for me about the teaching does, apart from the fact that students ask naive, intelligent questions, which is also very enlivening. Things you've got used to and sort of accepted, they will challenge, and that's important too. So I think I've talked long enough. I'll answer any questions you want, if anybody has any.
Richard, why don't you describe the situation and why the trade between you and Phil?
Oh, that was Hughes. Hughes didn't want me to be chairman. And I fought them for a couple of years, and then they said, well, you've got to choose. Either you're chairman or you have a Hughes position. Wasn't hard. So we engineered the switch because they didn't mind me being head of the cancer center. I did that for 10 years. That was a job they felt one could handle as well as being a Hughes investigator.
I was Dean of Science.
I remember you were, yes.
I had quite a hard time convincing Phil.
I remember that too. I wasn't going to go into that discussion, but that's why I used the word engineered. But it worked, because both of us knew what the other side of the street was about, and actually it worked very well. So.
Richard, you made, I think, a
microphone, please.
I'm sorry?
Can you use that?
Oh, can I use the microphone? Yeah. You made it what I think is a very profound point that I've made also, the importance of teaching in a research career. Do you have any specific examples from your own career of how teaching actually helped your research?
Yeah, I can give you one very specific example. So in the late 70s, David Botstein and I put together the current version of 750, which came out of, I'll do a digression and then answer your question, came out of a faculty dinner where we were having an argument that you couldn't teach rigor with eukaryotic systems. That's what the microbiologists said and the biochemists. You've got to teach rigor with prokaryotic systems and biochemistry. Eukaryotic biology is too fuzzy, you can't teach rigor with it. And David and I were heatedly arguing the other side of that argument. And then this was, I think, Malcolm Gefter said, okay, do it. So we decided to restructure what was then mostly a molecular genetics course into what's now 750. And we had this idea that there were 12 big questions in biology, because there are 12 weeks in the term, and that we would set up 12 sessions where we addressed the same sort of question in a prokaryotic system and a eukaryotic system in parallel, so that you could see that you could apply the same logic, that's where the title came from, the same logic but different methods to answering the same sort of question. We never really could come up with 12 big questions like that, but some of them are still there. Complementation is still there. It was Benzer and some eukaryotic experiments, which were other sorts of complementation. The idea, you can test lots of things by complementation. The logic's the same, whether it's genetics or whether it's biochemistry. Anyway, so David and I taught this course for several years together, and we used to have dinner every night before the course, which started at 7. And we used to go to the F&T around the corner, some of you will remember it, and have dinner and figure out what we were going to pitch at the students after dinner, you know, how we were going to structure the discussion. And then we talked science. So David and I talked science a lot. And at the time, we were beginning to think about cloning fibronectin, it's big. It was really hard to clone. And I spent a lot of time talking to David about how to do it, and got a lot of stimulus from him that it was a worthwhile thing, a feasible thing, and I think that motivated us to jump into cloning of so many of the challenging molecules of cell biology, big ones, right, that were not easy to clone in those days. So I'm not sure we'd have done it without that teaching experience with David. So, David's done lots of other things to influence my career in subsequent years. We really had a great time teaching that course, and we used to teach courses at Cold Spring Harbor together. And those sort of collegialities that you get from teaching with people, they're like the collegialities you students get from living in the pit together. People you know in the pit, you'll know for the rest of your life. So, people you teach with, you know for the rest of your life. So. No more questions?
It's a surprisingly hesitant group. This guy has a real interesting career. Do you want to talk, I mean, something you've avoided talking about, running the stem cell program for the National Academy?
Yeah.
You want to talk?
Well, I guess I sort of alluded to the politics in the 60s and now. I think that one shouldn't, and I certainly wouldn't be fully satisfied by just research and teaching. And I actually think one of the things that MIT is very good at is community service. It's encouraged here. And I got into the stem cell thing through the Cell Biology Society, which has a big public policy activity influencing, trying to influence Congress and educate Congress to support science. And I got into the stem cell thing by testifying at the Specter-Harkin Committee in favor of stem cell support for stem cells and legislation to support stem cell research. And that was a lot of fun, actually. I testified with Mary Tyler Moore and Michael J. Fox. And they had us in the back room before we went in there, and we were all nervous, but they were really nervous. And I thought, my goodness, these people, these are professional actors. Why are they so nervous? And then we got in there, and they were spectacular. Once they were with the microphone and an audience, they just switched on. And you know, my performance was pitiful. Theirs was spectacular. So I had been lobbying for stem cell research, and the Academy asked me to chair a committee because the government was doing nothing about it. It was basically avoiding the issue, and there was no regulation at all. And it was kind of like the recombinant DNA period. There's a parallel there. The scientists decided, in this case the National Academies decided, that the scientists should regulate this science if there were any issues to be addressed. The scientists knew them best and should write the guidelines. So they asked me and some other people to do it. Bob Horvitz was on that committee too, and we wrote in 9 months the guidelines which now are the principles by which stem cell research is run in this country. And they've effectively been adopted into law by some states, not yet by the government, although my prediction is after the election that's probably what will happen. They'll pick up those guidelines and turn them into regulations. So I think that sort of thing that scientists can do to guide the way science is done, to educate Congress about science and the public, is another really important thing to do. And it's very rewarding, actually, discovering that the government is influenceable. So. Yep, Herman?
Richard, you had a unique experience with science education in England and here, in this country, especially at MIT. I spent about 6 months in Oxford many years ago, in the lab, Rod Ford's lab actually, and one graduate student who worked as an apprentice with Rod. That was about it. May have taken some courses, but no one paid much attention to that. I also came from a medical school background, where graduate education was pretty much limited to the graduate student being an apprentice in a lab, in some preclinical lab. At MIT, when I came here, the experience was totally different. It's a highly organized, well-organized program. Students take courses, terrific courses, many of them, then they do the lab work and research. So my question is, given those big extremes, way of carrying on graduate education, in terms of the final output, does it make a difference?
Yeah, I think it does, and I think I was really lucky to do it in the order I did it, because I think the undergraduate education was much better there than it is here. It was more advanced, you were more on your own, you were given stuff and you could explore it to your heart's content or ignore it. We always had choice on exams, and I wasn't that interested in the polyphenols that color tea, and so I just ignored that bit of the biochemistry class and concentrated on the bits I like, as an example. And then I was really lucky to come to MIT, particularly to MIT, but partly deliberately to come to the States, because I knew there were courses at the beginning in the States, and there weren't in Britain. And I still wanted to do courses. I knew there were lots of things I didn't know, and I loved the courses. And actually, we took many more courses than the students do now. We took courses well into our second year. As I said, prelims was a much bigger deal. It went on for a week, the second part of prelims. It was an oral exam, 2 written exams, a take-home paper. So we did more coursework then, and I actually think it's a pity we don't do as much now. I think the students miss out a bit, and I encourage them to take a course or 2 in their later years. And that was partly why I came, because I wanted that experience. It meant it took 5 years to get a degree here, whereas it would have taken me only 3 in Britain, but I thought it would be a better degree. And I still think that's true. And in fact, the British graduate schools have now moved to a 4-year program, which I think is about right, because when people finish college in Britain, they're a bit more advanced than when they finish college here on average, because they're more advanced when they finish high school, because they don't waste so much time in high school as they do in American high school. So if to pick the best of all worlds, I think go through high school and college in Britain and then come here for graduate school, which was what I did, and I was lucky to do that. So.
Richard, you were president of the American Society of Cell Biology, and that led you into the stem cell effort.
Yeah.
Can you say something about the role of scientific societies and what opportunities you think there might be for younger people to be involved?
Okay, good question. Scientific societies obviously run meetings, scientific meetings, and that's an important part of what they do. And those are opportunities for people to present posters and talks, which people should take. But they also do other things, and which is what I think Bob is alluding to. That society in particular, but it's not unique, has a lot of career advice type things. Sessions at the annual meetings that illuminate how can you get into politics. If you wanted to become a congressional staffer, how could you do that? If you wanted to go into industry or journalism or any of the many things you can do with a science degree, that's a good place to find out about those things, and they're also spelled out in the newsletter, the monthly newsletter of the Society. The Society also does a lot in public policy. It was probably one of the leading societies. Others have followed, but I think it's probably true that that society led the charge on this, to recognize the other aspects of the society, education, primary school, K through 12 education, generating teaching materials, recognizing people who do a really good job in teaching at the school level, bringing in senators and congressmen and their staffers to the meetings to have them talk to the scientists about what it means to run the government and get feedback from the scientists. So I think that broader world out there, the scientific societies are a really good way It's not that none of that is done here, some of it is done here, but the societies also have a big activity in that way. And as I said, it's very rewarding to get engaged in the way, we're all paid by the taxpayer to do what we do, and we keep asking them for more money. Well, they need a good case for why they should give us more money. And to learn from the Congress people what it takes to, what else has to be given up if you're going to give more money to NIH, and what the case needs to be if we want to persuade them, as we believe, that more money spent there would be well spent. They have to take it away from somewhere else. So finding out what it is that they need to know, they're actually very grateful for the help, because when they want to advocate for support for NIH, let's say it's Kennedy's office, who's a big supporter of NIH, he needs material, he needs information that he can add to that argument. I remember when the cases against the tobacco companies came in, and there was all that money from that settlement, how was it going to be spent? And the federal government had some of that money, the states had some of it. And I remember going down to the Capitol Hill to meet with Kennedy and his staffers about the bill they were writing about the spending of the tobacco money. And it had no peer review in it. The money was going to be doled out by some sort of committee. And we thought that was a bad idea. It ought to be done by peer review. So we went down there to persuade them of that. And it turned out one of our former graduate students was on his staff and was actually writing the bill. So we went out in the hallway with her and wrote the clauses in about the peer review. That bill never passed, actually, but if it had, we'd have had a big influence on how the money was dealt out in an important way, I think. So, and they love it when students go to visit. So ASCB and other societies, there's a program called the Coalition for Life Sciences, which is a sort of inter-society public policy group on which Bob and I both sit. And they run Capitol Hill visits, and some of those are for students. And it's a great experience to go down there and be educated about how Capitol Hill works, get into the offices and talk to some people, and they love to hear from young people. They do a better job than we do. So. Time to go. Oh, yep.
Granted, there are still 12 weeks in a semester. What are the new burning questions in science?
Oh, that's a tough one. Well, there's a series of neuroscience questions I would say are right up there. I mentioned that cytoplasmic architecture question that was posed then. It's a really fascinating question. How do you inherit cytoplasmic structure like that? And it appears in more subtle ways than it is in protozoa, so I'd add that one in there. Aspects of evolution, I think, are much more accessible than they were, given the genomic approaches that you can now apply to them. So, I mean, molecular biology was a problem, and then it was a tool. And genomics was a problem, and now it's a tool. And applying that tool to some of the disease questions, I don't think those are big questions in the same way as some of the other ones, but they're things that, I must say, in my science, the fact that what we do has a bearing on drugs and the market, not directly, but indirectly. I mean, I don't do any of that, but the fact that other people do it with the science that we do, I find enriches my feeling about it. MIT in the 60s and 70s was an ivory tower. It was not applied, explicitly not applied. This was pure science. And when I went to ICRF, and it's a cancer institute, one of the things we did was set up some postdoc courses. There were some MDs there who were there to learn how to do science. And we got them to teach us cancer, the pathology of cancer, the epidemiology of cancer. And I think applying genomic technologies to some of those issues, which are still there, something perhaps we should spend a bit more time teaching. Infectious disease, those are more applied, but I think they're really interesting and really important. Vaccinology, interesting bit of, the latest version of Immunological problems, how do you make a decent vaccine to malaria? Still not solved, or to AIDS. But I think the big questions are neuroscience. The big basic questions, I'd say, are neuroscience.
Okay. I think with that note, we should thank Richard for a wonderful conversation.
Thank you.